Hello everyone and welcome to the video. Today we are going to talk about one of the most important topics in industrial biotechnology, the bacterial growth cycle and the requirements needed for bacteria to grow. So without further ado, let's start by bacterial growth. Bacterial growth doesn't refer to the increase in the size of a cell. However, bacterial growth refers to the increase of the viable cells inside a population. Bacteria grow through binary fission, so a single cell duplicates its genetic components, then duplicate into two daughter cells. Both of the daughter cells contain the same characteristics from their mother. Bacterial growth cycle can be classified into four different phases: the lag phase, log phase, stationary phase, and the death phase. Let's see this graph. We are going to draw the logarithmic of living bacterial cells on the y-axis and the time on the x-axis. The first phase in the microbial growth cycle is the lag phase, in which bacteria adapt to the new environment. There is little to no cell division, but the cells are highly active, synthesizing enzymes, RNA, and other substances necessary for growth. Then we move on to the log phase, in which cells are dividing rapidly and exponentially at their maximum rate, producing the characteristic steep straight slope on a logarithmic growth plot. After a period of time, we reach stationary phase, in which the rate of cell division equals the rate of cell death. This occurs because essential nutrients become depleted and toxic waste products accumulate. Finally, we reach the death or decline phase, in which the rate of cell death exceeds the rate of cell division. The number of viable cells decreases exponentially as the environment becomes too harsh to sustain life. Bacterial growth has two requirements: physical requirements and chemical requirements. The chemical requirements provide the building blocks for the cells, and physical requirements provide the environment in which reproduction can occur. Let's see some examples illustrating the chemical and physical requirements. First of all, carbon source. Carbon is the fundamental element for all organic molecules, including structural components such as lipids, proteins, and energy storage. Bacteria are classified based on how they obtain carbon into autotrophs and heterotrophs. Autotrophs utilize inorganic carbon such as carbon dioxide, and heterotrophs utilize organic carbon such as carbohydrates. Most of the pathogenic bacteria are classified as heterotrophs. Another example is the nitrogen. Nitrogen is very important for synthesizing amino acids. And now let's go and talk about one of the most important factors affecting bacterial growth, which is the oxygen availability. Consider this test tube. If you inoculate a population inside this test tube and you kept the lid open or keep it loose, and we watch what is happening inside the test tube. If the population growth occurs at the top section, then we classify this bacteria as obligate aerobes because they require oxygen to live. However, if the population growth occurred at the bottom, then we classify them as obligate anaerobes because they are killed by oxygen. If bacteria seems not to care whether there is oxygen or not, then we call them facultative anaerobes. And finally, if bacteria tolerate oxygen but do not use it, we call them aerotolerant anaerobes. Now let's move on to another important variable, which is the temperature. Bacteria can be classified into psychrophiles, mesophiles, thermophiles, and hyperthermophiles. Psychrophiles can live in cold environment, mesophiles can live in moderate environment, thermophiles in relatively high temperature environment, and hyperthermophiles in a high temperature environment. Now, after studying the bacterial growth cycle and knowing its distinct phases, we need to answer the question, why biotechnology can overcome chemical technology? Biotechnology work under mild conditions, and it doesn't have toxic byproducts. So it's considered more economically friendly and sustainable. If you're interested in biotechnology and want to know more, make sure to subscribe and to follow this channel because we're going to have more topics regarding this topic in this playlist. Thank you so much and see you in the next one. Goodbye.